I'd like to start by thanking Dr. Curl for that very kind introduction. And as a side note, no, you did not ruin my talk, so no worries about the computer. So it's a real privilege to be here with you today, to exchange ideas with many distinguished leaders and innovators in so many disciplines. And I can't tell you how humbling and how inspiring it is to accept the Edith and Peter O'Donnell Award. On behalf of my team and our collaborators, and of course my family, I want to thank the committee for all their hard work. What I hope to do for you today is to share some of our team's recent advances over the past decade or so in developing new genetic technologies, and then how we've been leveraging these tools to discover new vulnerabilities, new addictions, if you will, that can be developed into cancer therapeutics. So with the rapid developments of genetics and genomics research, it's clear that the science of studying and treating cancer has entered into a time of both profound potential and also some daunting obstacles. And one can see this exciting crossroads with really just a brief glance at how we got here, studying the relationship between genes and cancer. Now, it was over 100 years ago that the German biologist Boveri proposed that it was the inappropriate function of our genetic material that played a role in cancer. But it was really a series of discoveries from the 60s to the 90s where the fields of genetics and cancer would become intimately and forever linked. For instance, in the early 80s, several prominent laboratories almost simultaneously discovered the first human oncogene, showing that tumor cells harbored a mutant gene, that one could transfer that mutant gene into normal cells and transform them into malignant derivatives. And then 2 decades later, the era of targeted therapies and precision medicine, if you will, was ushered in with 2 drugs, Herceptin for breast cancer and Gleevec for a lethal form of leukemia. Now, of course, this audience is well versed with what was special about these drugs, that they targeted the very mutated cancer genes that were driving the tumor and demonstrated conclusively that human cancers were sometimes addicted to their mutated cancer genes. Now, since the completion of the first human genome sequence map about a decade ago, we've seen a true revolution in the mapping of aberrations in cancer genomes. And a decade later, one can see that the results have been absolutely staggering. So over 2 million mutations in the cancer genomes we've sequenced thus far, affecting nearly 30,000 human genes, with 600 or more of these postulated to be potential drivers of cancer. And yet, comparatively, only a handful of cancers have really been impacted in any meaningful way by drugs that target such cancer drivers. And this inadequacy of cancer medicine really hit home when my father was diagnosed with a lethal form of cancer. Now, there was no targeted therapy to offer him. Instead, he received chemotherapy that patients like him have been receiving for over 40 years. And to this day, we still don't have a satisfying genetic answer for why he had cancer. Now, what's obvious and I think dissatisfying to most of us in the cancer field today is that this is really the norm. That with the wealth of genomic data that we've amassed, we still don't have the genetic smoking guns, if you will. The causative genes underlie most cancers. So that brings me to my central focus of our laboratory. First, to delineate the genetic underpinnings for those cancers that we don't have sufficient understanding. And second, we're very interested in developing new principles for finding the key weaknesses of cancer. Now, these on the surface are very simple questions, but of course the answers are very difficult. And part of what makes them so difficult, and part of what Dr. Curl alluded to, was that we don't know what most of the genes, 90% or so, in our genome actually do. And so this is a major obstacle not just for cancer research, but also for most disease-related science. So how do we find the genes that are underlying diseases like cancer in an unbiased way? So in basic biology, and I am indeed a basic biologist, one of the most powerful ways that we discover the genes controlling an important process is called forward genetics. Now, forward genetics, or often referred to as genetic screens, is an approach in which genes are experimentally manipulated or perturbed in an unbiased manner. And then one looks for outliers, for situations where the gene alteration leads to changes that one is interested in, like cell growth or metastasis. And essentially, this approach allows one to find genes that are governing a critical process without a priori knowledge of what a gene does. So this type of approach has been a mainstay, really, in model organisms from yeast, fly, shown here, and worm, and has told us much of what we know about the world of developmental biology, signaling, and even some diseases. But a major limitation in disease-related research has been that such forward genetic approaches were, in large part, limited or not possible, and that's especially true in human cell systems. But that changed really with the watershed event or discovery of RNA interference by Craig Mello and Andy Fire, shown here. Now, RNA interference, or RNAi, is a mechanism used by many organisms, including man, to govern gene function. And at the center of this fascinating process are small interfering RNAs, shown here in blue. Now, these siRNAs, upon Watson-Crick base pairing, can destroy a messenger RNA in a very sequence-specific manner, thus interfering with the function of that gene. Now, there are many areas in biological systems where this process is key, but from an experimentalist point of view, like myself, what is key is that the siRNA destroys or impedes mRNA in a very sequence-specific manner. And thus, one can create artificial triggers that interfere with any mRNA and therefore any gene by simply changing the sequence of these small RNA triggers. So as the first RNAi publications started rolling out, we saw this as an opportunity really to capitalize on this burgeoning field. So with my postdoctoral mentor, Steve Elledge at Harvard, and our longtime collaborator, Greg Hannon at the Cold Spring Harbor, we sought to turn human cells essentially into a tractable system for genetic screens. And we did so really with two innovations. First, we created whole genome libraries of RNAi triggers that target all the protein coding genes in the human and mouse genomes, essentially engineering these triggers into retroviruses to enable screening of diverse human cells and tissues. Now, the impact here is that this is enabling us to do genetic screens in all human genes in an unbiased manner. The second is that we learned from our colleagues in the E. coli and the yeast fields and employed a barcoding system that enables us to track how each RNAi trigger affects a cell, but in a very large and complex population. So the net result here is that we've generated many generations of genetic tools that enable us and others in the community to screen for genes that play an important function in many disease states. And for us, that's cancer. So using these type of genetic tools, we've been focused much of our attention on cancers that are poorly understood. And the two vignettes that I'm going to share with you today stem from our efforts to understand an aggressive subtype of breast cancer that was so nicely introduced by Kent a moment ago. So this disease afflicts nearly 250,000 new women each year, or is diagnosed new in about 250,000 new cases each year. It's metastatic, often progressing to the lung, bone, and brain, as shown here. And that's a point I'll return back to in just a moment. Now, in contrast to HER2-amplified disease that you heard Kent so eloquently describe, this disease does not have a major mutated oncogene that has led us to new therapies. And indeed, it's a lack of targeted therapies is probably due in large part because we have such a poor understanding of the molecular etiology of this disease. So one of the major goals for our lab has been to use forward genetics to uncover many of the genes controlling triple-negative breast cancer. So in the first series of screens that I'll tell you about, the premise of the screen is really quite simple. We're searching in an unbiased way for genes that restrain the development of breast cancer. Now, the details of these screens are largely published. Essentially, we've engineered normal mammary epithelial cells from a healthy patient with a genome-wide RNAi library and search for genes whose loss of function would lead to breast cancer in experimental settings. Now, because of the barcoded RNAi technology that we developed, instead of the one gene, one postdoc approach that has been so long employed, we were able to perform the equivalent of about 500,000 genetic experiments within 2 months. Now, from these screens, we've discovered really an entire circuit of new genes that work together in governing the development and the progression of triple-negative breast cancer. What I'll describe briefly over the next few slides is how our exploration into one of these key breast cancer genes, P2PN12, is providing us some new ways to understand and possibly treat triple-negative patients. Now, as shown here, inactivation of P2PN12 by RNAi or any other mechanism really leads to the development or the malignant conversion of memory cells into cancer-like derivatives. And virtually every way we inactivate or perturb this gene, whether it's in human cell systems or mouse models now, inactivation of this gene leads to breast cancer and metastatic progression. Now, PTPN12 belongs to a class of enzymes that are very poorly understood and largely understudied. Intriguingly, we found this gene to be inactivated in nearly 70% of triple-negative breast cancers, making it one of the most frequently compromised genes in this malignancy. So we sought to understand the mechanisms by which this gene so potently and so frequently suppresses human breast cancer. So to understand this mechanism, a little context is needed. So please let me remind you that decorating the surface of every cell in the human body are receptors that are crucial for cell-to-cell communication. Now, these receptors receive information when they're bound by an extracellular ligand, and that induces them to be active. And they transmit this information into the cell via cascades of signaling proteins that ultimately instruct the cell's behavior in a process like proliferation, tissue specification, et cetera. Now, I'm presenting this paradigm as a very simple linear pathway. We all know that biology is instead very complex, and the specificity in a cell's decision-making is in part achieved because there are many such receptors on the surface, each receiving their own instruction. Now, in studying the mechanism by which PTPN12 suppresses breast cancer, we discovered that this protein actually serves as the primary off switch for a very select set of receptors on the cell surface, preventing them from remaining in the on state. Now, intriguingly, each of these PTPN12 regulated receptors has been found to be hyperactivated by mutation in other types of human cancer. But I would like to stress these receptors are not mutated in the context of human breast cancer. So this led to a very simple but important model for how PTPN12 restrains triple-negative breast cancer, specifically that it suppresses the malignant state not by antagonizing any one protein or substrate, but instead by constraining the function of multiple receptors in aggregate. And that loss of PTPN12 would effectively remove the off switch, leading to hyperactivation of this set, the specific set of receptors that in concert drive disease progression. Now, this model makes a very strong prediction that cancers with defects in PTPN12, which, as I told you, are very frequent in triple-negative breast cancer, may be dependent on these unleashed receptors, but in a combined fashion. So the implication here is that the inhibition of any one of these receptors would not yield a therapeutic benefit, but instead it's the combined inhibition of these receptors that would significantly impair triple-negative disease progression. So to test that important prediction, we turn to our collaborator, Mike Lewis, who for the past 8 or 9 years has been generating patient-derived xenografts of human breast cancers by essentially transplanting these models, these tumors, into the mouse mammary gland. Now, these PDX models, as they're called, serve as an important testing ground for new therapeutic ideas. And with Dr. Lewis, we essentially tested this therapeutic strategy by using 2 drugs, Crizotinib and Sunitinib, that inhibit these PTPN12 regulated receptors only when used in combination. Now, shown here is one such PDX model from a triple-negative patient. And as you can see, this drug combination results in an exquisite effect on this patient's triple-negative breast cancer, causing durable tumor regression to what's not shown here out to past 6 months. Now, what I'm not showing you here is that either drug alone has little to no effect against this aggressive tumor model. Now, this was a very exciting result for us, but like other cancers you've heard about today, triple-negative disease is a very heterogeneous malignancy. And so to test the effects of this new therapeutic strategy across such a heterogeneous series, we need to do the same experiment, essentially, in models from many triple-negative patients. And to date, we've tested the strategy in models from 16 independent triple-negative patients, all of whom had chemo-refractory triple-negative disease. And as one can see, without treatment, these breast cancer models are universally lethal to the animals within about a month's time. In contrast, treatment with this combination of receptors led to stable disease, or more often, complete regressions in nearly 70% of these triple-negative patient models. Now, for a patient population that has no effective targeted therapy to choose from, this was, I think, a remarkable result. And so we've now joined forces with our clinical colleagues at the Baylor Breast Center, Ken Osborne, Matthew Ellis, Mo Ramawi, and others, as well as our colleagues at Pfizer to bring this new therapy to patients, with a clinical trial currently ongoing. Now, We're very excited about this new trial, of course, but this therapeutic strategy, if it's successful, brings to light, I think, a broader potential. And that is the following, that receptor inhibitors may have at least 2 broad applications in the cancer clinic. Now, in the current paradigm, genetic mutation of the receptor, as you heard Kent describe, HER2 amplification in breast cancer, EGFR mutation in lung cancer, et cetera, leads to a dominant effect of the receptor in this malignancy. And evidence from the clinic suggests that extinguishing such a receptor provides at least some therapeutic benefit. Now, as I mentioned, most cancers do not actually harbor such receptor mutations, and triple-negative breast cancer is in this class. However, our data suggests that many cancers, like triple-negative disease, may lose the negative feedback from P2PN12 or other members of this phosphatase class of enzymes, and that unleashes the activity of perhaps 2 or more receptors in concert. Now, certainly I'm not standing here today suggesting that combining receptor inhibitors or kinase inhibitors is a new idea. Indeed, many such trials have been proposed, and unfortunately, most have failed. And I believe what's often been missing is really a fundamental understanding of how these receptors are coordinately regulated, or in this case, coordinately dysregulated. So this model suggests that defining such mechanisms of how receptors are coordinately dysregulated could open up a much larger swath of cancer patients that might respond to such receptor inhibitors that we have already in the clinic. So that brings me to my next vignette, which I think starts with a very simple question. What are the unique vulnerabilities of human cancer? Now, though cancer is by its nature a disease of aggressive cells, we wondered if, much like the Greek warrior Achilles, that cancer would have key and unexpected weaknesses that could be leveraged for new therapies. Now, a major area of focus in my laboratory has been to search for these weaknesses by understanding the stresses that cancer cells experience. Now, that may sound a little counterintuitive, but the field has long recognized that tumors are constantly under a barrage of stress. Some of these stresses are extrinsic, whether it's a lack of oxygen and nutrients as they grow, or, as we heard so beautifully described by Dr. Allison, attack from the immune system. Certainly, many of these stresses are extrinsic. But some stresses are intrinsic to the tumor cell, like metabolic rewiring or issues in DNA repair. And we've been largely focused on these stresses that are intrinsic to the cancer cell. Now, you might fairly ask, where do such cell-intrinsic stresses come from? And a common source of these stresses may actually surprise you, and that is that these stresses are often conferred by the oncogene or the cancer-causing gene itself. Now, much of the cancer community focuses on understanding how these mutations in cancer genes leads to the behaviors underlying cancer, like unlimited growth and proliferation. However, it's also become clear that hyperactivation of many such cancer drivers also elicits heightened or new stresses in cancer cells. Now, if this is true, it has some pretty obvious implications for cancer therapy. One can imagine that exacerbating such tumor cell stresses on the right side of this balance may provide tractable and fertile ground for developing cancer-selective medicines. Now, it's in this conceptual context that we've been searching for, A, what types of stresses are conferred by oncogenes, and B, what mechanisms do tumors and tumor cells use to either evolve or to tolerate such stresses. Now, in the last few slides, I'd like to share some of our work delineating the stresses and weaknesses conferred by a very old oncogene, and that is the Myc transcription factor. Now, Myc is one of the first discovered oncogenes, and its mutation or aberrant activation happens in about 1/3 of all human malignancy. And typically, it drives some of the worst diseases. Now, because of its prominent role in cancer, a whole field in cancer biology has been devoted to studying this oncogene for over 30 years. And yet, we still don't have a tractable therapy that can inhibit this key cancer gene. Now, while Myc endows cells with many malignant behaviors, our lab is really more interested in trying to delineate the types of stresses that are imposed by Myc, and then whether we can discern or turn such pivotal stresses or weaknesses into points of therapy. Now, our starting point was very simple. We wanted to discover which genes are actually required to tolerate these Myc-induced stresses. We were going to let these genes essentially point us to new and interesting areas of Myc biology. Now, the premise of this genetic screen is very simple. We isolated normal cells, again, from a healthy human mammary gland, and then engineered them to express oncogenic Myc. So these two cell states essentially have, or they differ by really only one variable, and that is the absence or presence of oncogenic Myc. And then we perform screens for genes that are essentially have no effect on cells in a normal or wild type state, but were strongly detrimental when cells were experiencing oncogenic Myc. So in this screen, we performed essentially 600,000 independent genetic tests covering 30,000 genes and found some truly unexpected areas of biology. And what I'll share with you here for the last few minutes is really the big surprise that emerged from that set of screens. We found that a molecular machine in the cell called the spliceosome is critical for cells to survive the stress of oncogenic Myc. Now shown here is just some representative data. And as one can see, that in Myc hyperactive cells, inhibition of the spliceosome leads to dramatic effects on cell viability. And if one takes a closer look at these cells, one finds many processes essential for life to be all failing simultaneously. So what is the spliceosome? And why is it so pivotal for these Myc-driven cancers? So to put this question in context, first I need to remind you of just a couple of fundamental elements of molecular biology. Now most of our genome encodes messenger RNA, or most of the genes in our genome encode messenger RNA, shown here. And these genes, or these mRNAs, then of course are translated into proteins, which are the biochemical units that carry out many critical cell functions. Now the parts of the gene that actually encode for those proteins is marked here in green, and those are called exons. And these exons are separated by long, non-coding sequences within the gene called introns, marked here in yellow. Now, as these exons are separated by non-coding sequences, they need to be conjoined in some way to create a functional and usable messenger RNA. Now, key to all gene expression is that for the information to be transmitted properly, the pre-messenger RNA has to be processed to remove these non-coding introns. Now, the spliceosome is actually the molecular machine that carries out this process, removing these intronic sequences and conjoining these adjacent exonic sequences. Now I'll return to the question I posed to you just a moment ago. Why does the Myc oncogene make cells so exquisitely dependent on this cellular molecular machine? And for that, we were actually helped by a long-standing observation in the Myc community, that Myc-driven cancer cells consistently synthesize more pre-messenger RNA. And the mechanisms for this are very hotly debated even today, but the part or the phenomena we're most interested in is actually the consequence of this activity. The elevated pre-mRNA synthesis in Myc-driven tumor cells may actually burden the spliceosome, essentially increasing the need for optimal spliceosomal function within these cancer cells, and that would be needed to properly process and mature messenger RNA, and ultimately to supply cancer cells with all of their biosynthetic needs. So from a therapeutics perspective, this suggests a pretty simple prediction, that normal cells can actually tolerate perturbations in the efficiency of their splicing. But in contrast, tumor cells driven by Myc are actually addicted to optimal spliceosomal function. And in the absence of that optimal function, this creates a bottleneck of sorts. Many of these cellular mRNAs are now not properly maturing. And instead, this causes unspliced, non-functional mRNA to accumulate in these cells and causes a catastrophic burden on cells that ultimately results in cancer cell death. Now, this was certainly an unexpected, surprising addiction that we've discovered in Myc-driven cancers. And we've begun testing whether this very basic science discovery can be leveraged into the clinic by working with collaborators to develop and to test new drugs targeting the human spliceosome. And of course, we're doing that in Myc-driven human cancers. So as I mentioned previously, Myc-driven cancers, they're some of the worst. They're aggressive and very highly metastatic. And as you heard from speakers earlier today, it's really metastatic disease that is the great killer of most cancer patients. So shown here, we've allowed a Myc-driven breast cancer to form metastatic lesions into the lung of these mice. And then we've marked these tumor cells with a bioluminescent signal so that one can actually observe these metastases in living animals. Now, quite remarkably, administration of a newly developed spliceosome inhibitor for about a week caused a greater than 20-fold reduction in the burden of metastases in these animals. So this and other data support the idea that targeting the spliceosome is a tractable area, or at least a promising area for new therapeutics. And we're working with both colleagues in academia and in pharma to develop new burgeoning inhibitors. So to summarize this last vignette, we've uncovered a new type of cancer cell stress in Myc-driven malignancies. And this stress arises from really a pathologic pressure that this oncogene puts on the human spliceosome. But more broadly, I would contend that this area of delineating what types of stresses are conferred by Myc or any other given oncogene is an area that's ripe for exploration in both biology and in cancer therapeutics. Now, in my last slide, I return to the question I posed to you at the beginning of this talk. How do we tackle cancers that don't fall into the classic paradigm that Kent so well described of directly targeting a mutant cancer gene, of which triple-negative breast cancer is a striking and prominent example? Now, I've summarized for you 2 different classes of addictions that we've discovered in this challenging form of breast cancer. And I believe it's these types of addictions, as well as the approaches that we use to discover them, that may have a significant impact in other difficult-to-treat malignancies. Now, this is where being a part of the Texas Medical Center, the Baylor community, and of course the greater Texas community has been very important for us. Myself and my team, we're geneticists, basic biologists, and it's really only in the bed or in the community of Baylor and the greater Texas Medical Center that we've been so fortunate to try to leverage some of these basic science discoveries into new possible therapeutics. And with that, I'd like to thank many people. This is actually an incomplete list of all of those that have contributed to the work that I've shown you. Many senior mentors at Baylor that have done just a tremendous job of helping young faculty like myself. And I'd like to, of course, acknowledge our support in the funding agencies. And with that, thank you for your time and attention, and I'd be happy to take any questions.
So I think this is really nice work, and I thought the spliceosome finding in particular was really intriguing. And by the way, congratulations also. I have a couple questions about the spliceosome finding. Does it look like you're primarily seeing a general defect in splicing or a failure to correctly regulate alternative splicing? And were the components that you're finding primarily the protein or the RNA components of the spliceosome?
Those are 2 great questions. The first is actually, of course, in a short talk like this, I didn't have time to talk a lot about the molecular biology, but it turns out that Myc-driven cancer cells, it's not just a defect in alternative splicing that they're suffering from, it's actually optimal activity. So when we make even modest perturbations in the spliceosome, it is havoc across most of splicing. So we see a lot of intron retention, which of course would be a phenotype you'd expect. With regard to the spliceosome components, we've been doing this both genetically and pharmacologically, marching through, I didn't have time to say, but the spliceosome is actually over 130 proteins, just the core machinery. And we think many of those, at least 12 of those are enzymes, represent some very viable therapeutic entry points. But I'd be happy to talk more about the implications later.
That was a wonderful talk. I enjoyed it immensely. The basic idea with RNAi is that you knock down the RNA, and thus you've knocked down the levels of a protein, and you can figure out what that protein is doing. And one of the challenges is sometimes that doesn't work terribly well. Like, my lab has had experience where that doesn't work terribly well. I would guess that from your immense experience with this, you could maybe comment on the limitations of RNAi as an approach for looking at protein function.
Yeah, absolutely. So you raise an important point. We've been in the sort of engineering community of RNAi for about a better part of it, 12 years. And certainly there are many studies in the literature where people don't do a very careful job of trying to delineate whether the triggers that they're using are actually affecting the genes they think they're affecting. There have been issues with off-targets, issues with, of course, these usually create hypomorphs, not complete loss of function alleles. But I'd say that there are pretty rigorous secondary screens that one can do to kind of funnel your attention into things that are, what you might consider biologically interesting and real. Of course, I'm sure you're aware there are many different technologies now, the CRISPR-Cas and others that people are using that I think are important complements to how we view this sort of genetic engineering problem.
Thanks.
Yeah, thanks, Russ.